The overall goal of the following experiment is to determine relative changes in tRNA charging levels. This is achieved by isolating RNA at a low pH before and after treatment to isolate tRNA with intact charging levels. As a second step, the RNA is subjected to a per-iodate oxidation treatment, which allows for determination of relative charging levels. Next, ligate samples to a fluorescent oligonucleotide and hybridize on a microarray in order to measure changes in ligation levels due to tRNA charging. Results are obtained that show genomic level changes in tRNA charging based on tRNA microarray technology.
Hi, I'm John Zaborski from the laboratory of Dr. Tao Pan in the Department of Biochemistry and Molecular Biology at the University of Chicago. Today I'll be showing you a procedure for isolating and measuring charged tRNA at the genomic level. We use this to measure changes in tRNA charging during stress response in our lab. So, let's get started.
To isolate total charged tRNA, start by growing a minimum of the equivalent of 30 OD600 of yeast cells, which should produce 30 micrograms of total RNA. Pellet the cells in a tabletop centrifuge. Resuspend cells in a lysis buffer solution. Add an equal volume of sodium acetate saturated phenyl chloroform and vortex for 30 seconds, followed by icing for at least 30 seconds. Repeat vortexing and icing 2 more times. Centrifuge the sample at 18,600 RFC for 15 minutes at 4 degrees Celsius. Then remove the aqueous layer and perform another acetate-saturated phenyl chloroform extraction. After the second extraction, precipitate the RNA by adding 2.7x volumes of ethanol and centrifuging at 18,600 RFC for 30 minutes at 4 degrees Celsius. Resuspend RNA pellets in lysis buffer solution and then precipitate with ethanol a second time. Finally, resuspend the RNA in a solution containing 10 millimolar buffered acetate and 1 millimolar EDTA for subsequent treatment. The sample can be stably stored at -80 degrees Celsius for about 2 weeks. Longer storage is not recommended, as some charged tRNA will start to deacylate. Prior to labeling with Cy3 or Cy5 fluorescent oligonucleotide tags, the total tRNA is subjected to a per-iodate oxidation treatment, which allows for determination of relative charging levels. Mix the total RNA with 0.066 micromolar of each E. coli tRNA standard and 100 millimolar buffered acetate in the presence of 50 millimolar sodium periodate. For the control tRNA sample that will not be oxidized, use 50 millimolar sodium chloride in place of the sodium periodate. Incubate the reactions for 30 minutes at room temperature. After 30 minutes, quench the reaction by adding glucose to 100 millimolar and incubating at room temperature for 5 minutes. Once the reaction is quenched, perform a buffer exchange using a G25 spin column to remove any remaining sodium periodate from the samples. Precipitate the samples by adding buffered acetate to a final concentration of 133 millimolar, sodium chloride to a final concentration of 66 millimolar, and 2.7x volumes of ethanol. Next, deacylate the tRNA samples by resuspending them in 50 millimolar tris HCl and incubating at 37 degrees Celsius for 30 minutes. After 30 minutes, neutralize the reaction by adding an equal volume of 50 millimolar buffered acetate and 100 millimolar sodium chloride. Precipitate with 2.7x volumes of ethanol. Resuspend the RNA pellet in water at about 1 microgram per microliter. Once the control and oxidized samples have been checked for RNA quality by running on agarose gels, we can proceed with the fluorescent labeling. To attach fluorescent oligo tags onto the tRNA, combine 0.1 microgram per microliter of deacylated RNA with 4 micromolar Cy3 or Cy5 containing oligonucleotides, ligase buffer, 15% DMSO, 0.5 units per microliter of T4 DNA ligase, and yeast exophosphatase. Incubate at 16 degrees Celsius for over 16 hours. After ligation, mix the samples with 4 volumes of potassium acetate and potassium chloride, and then extract with an equal volume of phenyl chloroform. Following extraction of the aqueous phase, Precipitate the RNA preparations with ethanol. Finally, resuspend the RNA pellets in water to approximately 0.1 microgram per microliter. Prior to hybridization, boil microarray slides in distilled water for 1 to 2 minutes to remove unbound oligonucleotides. Next, combine the Cy3 or Cy5 labeled samples with 140 microliters of hybridization buffer, salmon sperm DNA to a final concentration of 140 micrograms per milliliter, and poly A to a final concentration of 70 micrograms per milliliter. Run a hybridization program on an automatic array hybridizer. We highly recommend the use of an automatic hybridization machine for tRNA work since manual hybridization produces high background. When the hybridization program is complete, manually wash the slides with Wash 3 solution by gently shaking in a 50 milliliter conical tube for 3 to 5 minutes. Wash the slides at least twice in tubes pre-wrapped with aluminum foil. Dry the slides by centrifuging at a low speed for 5 to 10 minutes. For optimal results, the slides should be scanned immediately after they are dried. Slides can be scanned 2 to 3 times without noticeable degradation in image quality. Representative results of a properly isolated total RNA sample and its analysis are shown here. This sample has a very clean spectrum, with an OD260 to OD280 ratio near 2, with no detectable protein or phenol contamination. When run on 1% agarose gels, both oxidized and control samples should show a strong tRNA band, with other possible nucleic acid bands varying between organisms. If a sample is noticeably weaker than other samples, or if smearing is observed, samples should not be used for microarrays. Microarrays should have a very low background, which is an order of magnitude lower than the weakest tRNA probe.
I've just shown you how to isolate and determine the relative charging levels of tRNA. When doing this procedure, it's important to remember to keep your reagents free of nucleases, especially during the oxidation and deacylation steps. Thanks for watching and good luck with your experiments.